everyone, welcome back to another video of STEMAC, and today we're going to talk about nucleus. So we're basically going to learn about the structure of nucleus and its basic functions. So we know that nucleus is present within our cells, right? So let's say this is a cell, and within the cell we can see that this is the cellular cytoplasm. Now the cytoplasm contains different types of organelles, and nucleus, this is the nucleus is included within those organelles. So the nucleus is present within this cytoplasm. Now if we zoom this nucleus, we can get a bigger picture, a clear picture of the nucleus here. So we can see that there are some components of the nucleus, different types of components. So at first we can see that this cellular structure within this nucleus is called the nucleolus. Now this nucleolus is responsible for forming or producing ribosomes, and surrounding this nucleus we can see some tangled thread-like or fiber-like structures, thin fiber-like structures, and this tangled structure is called chromatin. So we know chromatin contains DNA, right? And this DNA are coiled up together. And during meiosis or mitosis process, this chromatin can condense to chromosome. So this is basically the genetic material within the nucleus. So the surrounding both the nucleolus and the chromatin, we can see another jelly-like substance or structure. So this structure, this blue green dotted structure here is called the nucleoplasm. So the nucleoplasm is responsible for holding different types of molecules such as nucleotides or different types of enzymes, etc. So these are basically the inner components of a nucleus. Now we can see that surrounding these inner components there is another outer structure of the nucleus, and this outer structure is called nuclear envelope. So if we further zoom this nuclear envelope, we can see that here is a portion of the nuclear envelope, and we can see that the nuclear envelope has two layers, right? So the outer layer is called the outer membrane, and the inner layer is called the inner membrane. And this nuclear envelope contains two phospholipid bilayer within the nuclear envelope. So we can see that there are some pores within this nuclear envelope. So these are the pores, right? And we will refer to this pore as nuclear pores. So the nuclear pore, the function of the nuclear pore is to allow the transfer of different types of molecules into the nucleus and out of the nucleus. So let's say a nucleotide within the cytoplasm will come into the nucleus with the through the nuclear pores. So it is helping in the transfer of different types of molecules. Now one interesting fact about the nuclear envelope is that the outer membrane of the nuclear envelope has a continuous endoplasmic reticulum. So this is an endoplasmic reticulum, or in short ER. Now endoplasmic reticulum is an organelle, and it is of two types, which are smooth endoplasmic reticulum and rough endoplasmic reticulum. So in this case we're going to talk about rough endoplasmic reticulum. Rough. Now we say rough because there are some ribosomes attached to this endoplasmic reticulum. So these are ribosomes. Now, earlier we said that the nucleus, a nucleolus, is responsible for forming ribosomes, right? Now, these ribosomes can either move out of the nucleus and go to the cytoplasm, or they can either go and attach themselves with the endoplasmic reticulum. So, when there are ribosomes attached to the endoplasmic reticulum, we will call it the rough endoplasmic reticulum, and when there is no ribosomes present in the endoplasmic reticulum, we will call it smooth endoplasmic reticulum. So, ribosomes have a very cool feature, and we will see why. So, we learned that the nucleus contains chromatin, right? And chromatin contains DNA. So, DNA goes through a process of transcription. And with the transcription process, the DNA is converted to messenger RNA or mRNA. And this mRNA goes through another process called translation. And with the translation process, the mRNA is then converted to protein. So what happens is that when the DNA during the transcription process is converted to mRNA, the mRNA can move out of the nucleus, and the ribosomes can take this mRNA with them and bind themselves with the mRNA. So let's say there is an mRNA here, and they will bind with the ribosome. And what happens is that these ribosomes will take this mRNA and move it within the endoplasmic reticulum. They will move it to the endoplasmic reticulum. And within this endoplasmic reticulum, the mRNA will go through some modification, and then protein will be produced, right? So ribosomes are helping in the synthesis of proteins. So when the mRNA is converted to protein within the endoplasmic reticulum, they will then be released from the endoplasmic reticulum. So let's say they will be released from the endoplasmic reticulum in small vesicles. So this is a vesicle, and within the vesicle there is the protein that has been formed from the modification of the mRNA, and this vesicle will be released from the endoplasmic reticulum. And then this vesicle with the protein within them will then travel to Golgi apparatus for further modification. So this is the function of endoplasmic reticulum and ribosomes. So with that, our video comes to a conclusion, and hopefully with the video you can understand the structure of nucleus and its basic function better. Thank you for watching, and stay tuned for the next video.